So for the next maybe 40 minutes or so, I want to talk a little bit about modeling, and I only have very few slides. So the idea is that maybe we can do mostly a discussion, starting to talk about it now. Of course, we can continue it on Slack or tomorrow and the day after. So I assume you did watch the lecture, so I'm really just giving a few, highlights, what I think are sort of important points to emphasize. And then most of the time, hopefully, we can just talk. So when we say modeling, it's actually pretty broad, right? It's called, we're calling this within host modeling, and we have a certain kind of model in mind, but the point is that when you just say modeling, it could be all kinds of model types. And the ones that you're probably most familiar with, it, at least most of you, are sort of the statistical models, regression models and those type. So those are models too, but those are not the ones we want to focus on here. So in this module, we're specifically looking at models that are, basically implemented simulations. The hallmark is that they have mechanisms that of the system in an abstract form, which is different from regression models where you usually don't have mechanisms or processes. So here's a simple model that you've encountered in some of the lectures where we just modeled two entities, bacteria and immune response. And then on the right-hand side, you see these mechanisms in simple forms. Bacteria can grow, they can die, they can be killed by the immune response. So that is the hallmark of the kind of models we're looking at here, that you have models that have mechanisms, of course, in an abstract form, right? I mean, you've just heard Paul talk about all the complicated things the immune system has going. We can never model all of that in detail. So the trick is to get it just to the right level of detail that you can do something with say something useful with your model, but it's still tenable. You can do something with it, and especially you can set it up in a sense that is close to the system you're trying to model. And we're applying them here to within host, but I know a lot of you are familiar with these kind of models at other levels. For instance, at the epi level, something like the SIR model, that models a spread of pathogens on the population level. And then, of course, even outside of immunology or epidemiology, you have that in a lot of areas of science and engineering, those kind of models. Basically, when you know enough about a certain system that you can write it down in equations that have explicit mechanisms and processes, then you can use these kind of models. If you don't know enough about your system to do that, then these kind of models will not be very helpful. And so because a lot of you are familiar with these, what's sometimes called statistical models, I don't like that term too much, because the kind of models we're talking about here can also be statistical in the sense that you're fitting them to data. So I prefer the term non-mechanistic or phenomenological. Basically, you apply those models to data. Broadly speaking, you're looking for patterns in your data. And they can be very powerful. You can apply very sophisticated models to it. Classically with statistics, now it's machine learning and artificial intelligence, but it's all kind of the same idea. You have some sophisticated or simple model that you apply to data to try to look for patterns, and then potentially use it to, understand and maybe predict. The limitation is they don't describe the mechanisms that are going on that will lead to certain outcomes. So they kind of are, representation of the data, but in some sense always a bit black boxy because there is no specific mechanisms. And that's in contrast to the models we're looking at here, where you have to explicitly write down mechanisms that, produce the outcomes you're interested in. Of course, to be able to do that, you need to be able to know enough about your system that you can write down those models. So it's both the strength and the weakness of those mechanistic models. They give you more detailed information. They give you insights about mechanisms and processes, but you need to know at least something about your system to be able to write that down. And again, you can do those kind of models without data, and we'll actually look at that. We'll run a bunch of models. There's no data. We just analyze the model by itself. And then you can apply it to data, and that's when they become statistical. We'll actually not talk about that much in this course because it's just not enough time. But if you look in DSARM, the package, you will see there are several apps there that look at fitting these kind of models to data. So I'm pretty sure some of you will be interested in that topic. So then I suggest you take a look there. Broadly speaking, you can use these kind of models for different things. At the most basic one, you can just explore the behavior of a system. So if you have a system that you kind of know enough that you can build a model, but you don't fully understand the behavior as you change inputs, then you can build these kind of models and use it to explore what might happen. Then if you think that your model is reliable enough, then you can basically apply exactly the same approach. Is just to interpret the answers all differently instead of sort of a initial exploration. Now you can say, well, I actually believe this model captures reality pretty well, so I actually feel confident enough to make predictions. And then if you have data, you can much more rigorously test hypotheses, perform inference by comparing your models to the data and seeing how well they can match the data. Here's an example from a published paper 20 years ago now, where the authors built a model for TB within host. So I'm not showing the model here. It was intermediate complexity, definitely more complex than the kind of models we'll be looking at. And then they used it to explore and or predict what would happen if they knocked out certain cytokines out of their model. So in panel A, you see there are these different lines with IL-10, IL-4, IL-10, IL-12. These are always then in the simulation, in the model, knocked out. And then they were looking, okay, then what happens to other components in the model, for instance, bottom right, the total bacteria. So you can see there are two instances where the bacteria go down and one where they basically blow up, meaning that would be full-blown TB. So you can interpret that as exploratory, what would happen, assuming this model captures the system, or if you trust it, you could say, okay, we're predicting that if one gave a therapy where when depleted IL-10, we would see the following outcome. In this case, in this paper, they didn't fit it to data. But if you had the right kind of data, you could, of course, then compare your model predictions with the data. These simulation models are a certain kind of model, but within them, there's, again, a variety of kinds of models. And we'll mostly focus on what's called compartmental models. They're called that way because you're dividing the entities you're interested in this simple example, bacteria and immune response, into distinct compartments, and you're just tracking the total numbers. That makes them fairly simple. So you only need to track one variable for all the bacteria in the system. You just keep count of the numbers. It also matches most closely to what we usually measure in experiments, right? We just measure total number of T cells or total number of IL-10 or things like that. So they're most natural types of model to compare to the data we have. If you want to go more detailed, you can use agent-based or individual-based models or network models that are kind of a form of individual-based models often. There you model every entity or at least a subsection of the entities in a lot of detail. Those models are, of course, more detailed, so you can put much more realism into those, but that also makes them more complex. Here's an example, a very simple one where you basically had an area of cells, think of it maybe epithelial cells for flu. Those are the green ones, and then they get infected, that's red, and then there's virus, that's blue, and T cells that are white. And you can, for instance, study things here like spatial spread that you couldn't easily do in a compartmental models. Or you could give each one of these cells a different lifespan that you couldn't do in these other models either. So they allow for more realism, but it's much harder to compare them to data, and they are also harder to build and harder to run. So because of that, the compartmental models are still kind of the dominant kind of model being used, definitely on the within host level. I think at the epi level, we're a bit further in terms of being able to use more detailed models, but even they're still very data hungry to probably set them up. So that was all the highlights I wanted to go through. And so at this point, we really can spend the rest just talking, discussing. You can either unmute yourself and ask questions, or you can type them in the chat and I can read them. It can be about the modeling part, or if you have questions about the package, the R package, we can certainly discuss that too.
This is Marco. I had a question. So just quickly for the agent-based model with like in the within host context. So an agent would be like a single cell or a single like virus or single bacteria. Is that the way you're thinking about it?
Yeah. So at the most basic level, you model individual entities, in this case, individual viruses, individual bacteria. You can do hybrids. So for instance, you could model the cells individually, but you model the virus or the cytokines as a concentration that's sort of floating on top of that grid. So there are these hybrid approaches, and usually people take it because, you know, once you try to on a computer simulate every individual entity, things get really slow. So if you have to simulate 1 billion virus particles, it gets really slow. So then you might have to find some hybrid approaches. But yeah, so in general, that's what you think of. The agent is usually the individual unit. You know, sometimes you could think of in higher orders, you know, let's say you wanted to build a model, I've not seen one, but where you consider an agent or an entity to be a whole lymph node.
Okay.
That would be okay too. So, but the most basic thought is, you know, whatever is the fundamental entity, in this case would be cells. Now it gets tricky for cytokines, right? I mean, what's the fundamental unit of cytokines? A single molecule, I guess. But so then for those kind of things, usually people would probably model it as a concentration.
Okay. Then I have one more question, if that's okay. I was curious, like, have you ever seen cases where like you would link like a within host model to more of like a population or like individual based model where you're like maybe modeling the dynamics of an infection like inside a person, but then also linking it to like transmission between people, or is that like not really done commonly or at all?
Yeah, people started doing this in the individual based model. I've seen a few for flu where there's a sort of individual based model at the population level where they, you know, individual infected person, and then every infected person has their own little model running inside of them. And then depending on how much virus is produced in their inside model, that is how infectious they are in the population level. The main challenge with those is, again, computational power, because now you're trying to, you know, run model inside a model, and you have to pick fairly small time steps to make sure you capture the within host model well. So because of that, these sort of full-blown realistic ones aren't done too much. Often people do sort of hybrids or more than the compartmental type model. You know, let's say you have a within host model where you have the virus going up and down, and then you link that to a population level model where the rate of transmission is proportional to how much, you know, virus is currently produced by an individual. So there definitely people have been doing those kinds of models. Often you need to tailor it specifically to what the question is you want to answer, and you're trying to get away with the simplest version you can to answer the question you want. Just really full blown is still computationally very expensive, and you have to, you know, have access to a lot of computational power, and of course also you have to write a ton of code.
Yeah. So. All right, thank you.
So I had a question as well. So I know we're not focusing a lot about model data interface in this course, but so could you comment on the type of data that is generally available? Because the thing is, so in my personal work, I've come across data stream that are transcriptomic in nature. So that means they're not virus titers, but they're viral RNAs. And because the models are, so it's an indication of how much viral load there might be. But that is, to me, and when I think about it, it is not really the representation of how much infectious viral load there is in the system, because you can have waste RNA circulating in the, you know, so that's that one bit. And then there's the other bit, which is similarly, you have some idea about how the immune system is acting up as a function of the viral titer. but even that data is many a times just RNA or some sort of gene expression. So how do you sort of integrate that within a population level model? Or well, when I say population level model, I mean a compartmental model that models for numbers and not gene expression.
Yeah, so I mean that's where the modeling business becomes tricky, and there's no real recipe. I mean, usually it's then based on scientific understanding of the system. If you can say, I assume that RNA are proportional to infectious titer, and I know that the factor is about 10, or maybe it's changing during the infection. So that's really then, it needs to be informed by some kind of biological knowledge to be able to do that right. And that, in my opinion, really distinguishes good models from not so good models, is getting the biology right enough that the model is useful. I mean, you always have to make approximations, but doing them in the right way, such that your model is hopefully still capturing the essential features of you're trying to capture. That's sort of the tricky bit. And so, trying to figure out how can I use these kind of models with the data I have? Sometimes the answer is just no. Sometimes the answer is just, these kind of simulation models are not the right tool. And you might be at a level where, statistical and machine learning type models are the right tools. But then if you decide these are the right tools, then, you have to carefully think about how do I match the model representation to the data I have. So, I have virus in my model, but I measure those virus particles in, with the PCR or your tissue culture infectious dose or something like that. So, how do I map one onto the other? No simple recipe other than, careful thinking about how to do it, and hopefully, knowing enough about the system to be able to make a good approximation.
Okay.
I just wanted to chime in on that. So in the online video for the flu talk, we actually deal with different types of kind of measurement of infection, like you were just giving examples of. And so there's sort of two issues I think that you raised that are slightly different. So one is, what do you do if you've measured RNA versus infectious titer versus something else, and how do you if you're pulling from 3 different studies, how do you put those together? The other thing to keep in mind is that there might be technical problems with how they were measured, but they're really fundamentally measuring different things, and so you might want to feed them into your model in different places. And so the example that we give in the flu is measuring the number of infected cells versus the amount of infectious virus versus the amount of viral RNA. And all 3 of those numbers are not super well correlated and across all of the different infections that we look at there. And so there could be real biology that's different, and not just sort of kind of measurement error or technical variation that's driving those different numbers. And so, but I think that's something that's obscured a lot, because people just, in the text of the results, they just say, we measured virus levels or something, right? And you can measure that 10 different ways, and you really need to dig into the methods to see if they're comparable.
Sure. Good. Thanks.
Other questions?
I have a quick question, just kind of related to the first 2 that have been asked, partly thinking about these tools and particularly data streams. I know pathogen evolution for intra-host models is something that I'm a little familiar with, and I was wondering if there is a connection there, basically, when you're thinking about pathogen sequences. And because it seems to me that the intra-host evolution models are distinct from some of the more immunology-focused models, and I'm trying to think about whether there's any connection there.
Yeah, so classically, if you have sequence-based model, you do phylogenetic, phylodynamic type models, and historically they were not linked to the kind of models that we're looking at, where you kind of need quantities, numbers, how many of each. That's starting to change, especially at the epi level. There have been some efforts where people are trying to build models that have basically sort of a phylogenetic process, and underlying that is something like an SIR type model, and a mechanistic model of spread. And then if you had data on both, if you had data on sequence and if you had data on numbers, then you could combine those two. So that's really, in the last few years, only people have started to do that. And there have been some implementations. I think Beast has a module that starts doing things like that. So I think I haven't seen a paper yet where they apply that on the within host level, but I think it could be doable if you have the right kind of data. So, if you did, you probably would need to do deep sequencing, not just consensus sequence, and do that over time period inside a host. So you can have enough resolution if you want to do it during a single infection that you can get, some kind of evolutionary pattern. And then at the same time, you would measure load. Let's say it's a virus, you would measure viral load. And then you could potentially combine them. The tricky bit is that what's been developed so far is for fairly simple underlying dynamic models, like the SIR model, where you just, initially it's just an exponential growth model, right? But once it gets more complex, once you need to include all kinds of immune response pits, then I think the link is not there yet. People haven't quite figured out methodologically how to do that. But I think that's an area that we'll definitely keep developing, to trying to combine different types of data stream into models that can deal with both. Okay, yeah, Irene, your hand raised.
Yes, I had a question considering the parameters that normally explain the decay of one population. Can this parameter, for instance, belong to several factors? For example, I'm thinking of a scenario where you have a confection. Can this parameter represent the decline of this bacteria, of that pathogen, and also from another external factor?
Not sure I fully understand. Are you asking if sometimes you might have different distinct subpopulations and you need to distinguish between them?
Yeah.
Yeah, so, and you can do that with these compartmental models fairly easily. So, you can, let's say you had a compartment for bacteria and then you decided, no, actually, no, there are two distinct bacteria. Let's say there are some that are replicating and some that are more dormant. Then you can just introduce new equations, one equation for the replicating bacteria and one for the dormant bacteria. Okay. So, your model would get bigger because now you have to not just track total, but you would track them individually. But in principle, it's not hard to scale those models. Of course, once you start splitting things into too many compartments, it gets really complex. And at some point, if you want to get too detailed, you might just need to switch to these individual based models. But yes, you can certainly do that. Or let's say you had T cells and you had some T cells that you consider are the effector T cells and other T cells that are in exhausted state. So you could have two different variables for T cells, and you can, then each one gets their own mechanisms and processes. So yeah, if you have that kind of information and knowledge, then it might be worth including that into your model and making it more detailed.
Okay, thank you.
Okay, I see some on Slack. One question, why calling them simulation models? It's just another name, right? I mean, one could call them mechanistic models, but they're models that are, mechanistic that are not those. Of course, you could say these kind of models, one can also run them without simulating them. Though in practice, they're pretty much always simulated on a computer. So I don't know, this is currently my favorite terminology, but, some people call it mathematical models. You find just models, dynamical models. So the terminology is a bit loose, and for each term you could find some counter argument where you find another model that has that feature too, but it's not the kind of model you're talking about. So yeah, that's, I guess, the best I can say about the terminology. In practice, I think pretty much all of these models you simulate on a computer somehow. There's another question here. What we mean by compartmental models? Yeah, basically, when you track total numbers and put those into compartments or variables, so total number of infected cells, uninfected cells. Now, the way they're implemented then is quite often with ordinary differential equations, and those are mostly the models we'll be looking at, but it doesn't have to be. You could have equivalents that are discrete time. You could have stochastic versions of it. But the hallmark is that instead of trying to track individual entities, you're just tracking the total numbers of some type, total numbers of T cells, total numbers of IgG antibodies, total number of macrophages. So instead of splitting them. And then, as we just discussed, of course, you can split them a little bit. You could say, okay, no, I'm tracking total numbers of activated and resting macrophages. But still, your base is, you have a compartment for them, and you're just counting how many are going in and out of this compartment over time. So that's where that term comes from. Other questions?
I had another question if no one has anything else.
Go ahead.
I guess I was curious, again, I'm kind of come from the background of like having no within host modeling experience. So I was curious to know like what your data sources look like to try to parameterize these models. Like, are there publicly available data that you can just kind of like go to and grab from? Are you collecting data on your own? Like, what's that look like?
So for me personally, most of the data comes from collaborators like Paul or other immunologists, bench scientists. The NIH over the last maybe 10 or so years have made a big push towards trying to make these data more publicly available. And a lot of grants that are given these days have the stipulation that the producers of the data will have to publicly put them into repositories. So one very prominent one is called Import. That's one the NIH has had for a while. And basically what they're hoping or trying is to have something for, immunology, virology, microbiology data that's similar to GeneBank, where, someone puts it in, but then it can be reused many times. The limiting factor there is that it's still much more complex. The immunology or microbiology or virology data is much more tailored. So, and you need a lot of metadata, how exactly was this experiment done, what's going on here. If someone just gives you an Excel file with numbers in it, it's really hard to understand. So that has limited its use a bit, but I think it will keep going that direction while improving. So I've seen some papers now where people actually use data in Import and reanalyze it. And that's data that was not originally collected for that purpose. So, so there's more publicly available data. Now, the thing, of course, the good thing is it's there. You can just grab it. The disadvantage is if you have a question, there's no one to go to ask. Well, sometimes you can try to email the authors, but they might or might not get a response. If you have a direct collaborator, then it's much more likely you'll get an answer. So I'd say, there are publicly available resources that are starting to come. It's probably not as strong yet as, at the EPI level. But even there, if you want to get your hands onto some big cohort study or so, you might have to, contact the investigators directly. So that's basically it. But I would definitely, for anyone interested, check out Import. They have a nice interface. You can sort of search the data. The good thing is, the good and the bad, it's really raw data. So you can actually get the raw fax files with multiple of gigabytes or maybe even terabytes of data. But then often it's also in more processed form in spreadsheets.
Will it be possible to get a link for this board by any chance?
It's not the link, but I just type the title in, so just stick that in Google. I'm sure it's the first thing that will come up. Wait, hold on. That was a direct message to Cody. That doesn't help anyone other than Cody. Okay, try again here.
Thank you.
Another one from from Slack. I mentioned computational constraints, and if that suggests people should use TensorFlow. Well, TensorFlow, for those who don't know, it's sort of a fairly powerful artificial intelligence, machine learning tool, but it's completely different models. So, you know, those are these kind of statistical models that I mentioned earlier. And, you know, if you have complex data to analyze that, then those might be the right tools. But for the kind of models we're doing here, that just is not the right, you know, as you'll see quickly. Well, again, we won't really do the data fitting here, but if you go through the DSARM apps, you'll learn quickly that, you know, you can really only fit fairly simple models to the data, because otherwise your models are getting too complex and you just don't have the data to answer, you know, to estimate your parameters. So because of that, we have to keep our models still fairly simple. So, you know, a machine learning or artificial intelligence model that has, you know, thousands of parameters that need to be estimated, that just wouldn't work for the kind of approach we're doing here. Okay, and now as follow-up, should I start learning how to write code for GPUs? So that it gets technical, but, you know, for those not familiar, you know, graphics cards have actually become, GPUs have become quite powerful, such that a lot of stuff is actually not just done on the CPU of your computer, but on the graphics card. Unfortunately, the tools are still such that, you know, something like R doesn't have a nice time to just tell R, hey, no, instead of using my CPU of my processor, start using my GPU to run code. I hope that's coming. I would like to have more computational power. I'd always want more computational power. But currently the tools are still somewhat specialized enough and focused on these artificial intelligence and machine learning models that, you know, for the kind of models we're doing here, I haven't really seen them. So these big models I mentioned earlier, usually people run them on supercomputer clusters with thousands of CPUs, but I'm not sure if they're being run on GPUs. And then sometimes they also need to be written very customized, you know, they're writing it in C++ or something, which is much faster, but is also much harder to code in. So, you know, if you don't really need this, you know, and you plan on sticking with R because this is all good enough, and, you know, you don't want a more complex coding language, then, you know, you probably keep your problems simple. There's enough problems to be solved that can be done, you know, keeping things, models fairly simple. Another question here, how are you assessing whether your model complexity is justified to capture the biological hypotheses? That's a good question. It's a tricky question. And I don't think there's sort of a default answer. If you have data, then you can try to, you know, sort of fit your, and try to, if you're trying to estimate parameters from your model, you can try to fit it, and you can try to simulate actually your data and see if you can actually estimate your simulated ones. And again, if there's an example of that in the DSARM package, if you want to learn that more. Now, if your goal is not parameter estimation, or you don't have data, then, you know, usually you'd want to start simple, and you should build it based on what's, you know, what you think is justified, what you know about the system. You know, an example on how maybe not to do it is, I remember several years ago, I saw an HIV model that claimed it could predict the very slow decline of, you know, CD4 T cells over years. If you looked at the model, the way it predicted it, it just had something in the model that basically made them slowly go down. So there was no prediction, there was no mechanism, no biology in there. They just stuck it in the model, and sure, then that's what the model did. So that's not a good thing to do it. But, you know, if you have, you know, enough information to say, okay, there's a certain process or mechanism that I know is probably happening, or that I assume is happening, then you build a model, the simplest model you can with that information, and then you explore it and you see how it compares to what you know about the system. You can do that if you have the right data in sort of a statistically rigorous manner by fitting. But you can also sort of qualitatively compare. Let's say you know that for your particular system, after some time, the bacteria and the immune response reach some kind of steady state, some kind of stalemate. You build a model and you find that my model doesn't allow steady state. It's always just oscillations, or the bacteria always die out, or the bacteria always go through the roof and the immune system dies out. That tells you, okay, I might have not built my model quite right. There are some things I didn't quite capture. So it's kind of an iterative process. You start with what you think you know, you explore it, you compare to what you know, and then based on that, you keep building until, hopefully at some point you reach a model that you think is, fairly reasonable given the amount of data you have. And then you'll likely always reach a point where your model is good enough and you need further data to discriminate between different model variants. So that's kind of that iterative process. You push it, and then at some point you collect more data to, further, in some sense, it's always the important part is the ruling out, right? You can never show that something happens, but you can say, okay, I built this model and it doesn't agree with data, so I've learned something, I'm missing something, and then you keep building it. And at some point you might have confidence that, your model captures all the important stuff well enough. But again, there's no recipe. It's an iteration between, doing the mechanics of building the model and then using your scientific understanding of modifying it.